Hello, everyone. I'm Sean Eakins, the CEO and founder of Collaborations Pharmaceuticals. Thank you all for joining us around the world today for our presentation. Tom and myself will be giving the webinar today. In just a few moments, Tom will give a talk on using machine learning to repurpose drugs as antivirals for Ebola in silico to in vivo. We just have a few logistical points to point out before we get started today. We would be happy to take any questions, and you can do those in this particular software by clicking on the little symbol at the top of the screen. This icon allows you to write the questions into like a chat box. And then on the left-hand side of the screen, you'll also see that there is an attendee chat. So if you'd like to send any private messages, or if you just want to put your messages there, we'd be happy to answer them. We certainly still have a few people logging in, so we'll just be a few moments. I would like to give you a little bit of an outline on Collaborations Pharmaceuticals, introduce you to who we are. That will give us a few moments for a few more people to get into the system. And then Tom will give a presentation on the Ebola work that we've been doing for the last 5 or so years at the company. And once Tom has finished his presentation, I'll round off with a couple of slides. This will give you time to write your questions into the chat icon, and then we'll have the Q&A session. We'll go through the questions and we'll be happy to answer them. And if you have any follow-up questions, we'd be happy to take those as well. So again, those that have just joined, I'm Sean Eakins, the founder of Collaborations Pharmaceuticals. This is a small company that I founded in 2015. We're located in Raleigh, and we have 3 labs here and do BSL-2 level in vitro research, as well as computational work, which I'm going to tell you a little bit more about. We don't have any angel or VC funding to date, but we have been very fortunate to have been supported by over $7.6 million in grant funding from the NIH and Department of Defense. And we have a pretty robust molecule and services pipeline so that we do fee-for-service work with different companies. Our efforts so far have enabled us to file a number of patents as well as new molecule IP. And we've been fortunate to have 8 orphan drug designations from the FDA, as well as 2 rare disease designations most recently. So what do we do? As a small company, we focus very heavily on using artificial intelligence alongside my real drug discovery intelligence. I have over 24 years of experience in drug discovery, and we use our approaches really to develop clinical candidates for rare, neglected, and unmet therapeutic needs. We are essentially a drug discovery company with a contract services part as well. And we primarily use our machine learning technologies to help deliver results for our clients. We have a very heavy focus on early drug discovery, so from lead optimization through to candidate development. And we tend to focus primarily on very challenging ADME-tox type problems. We do a lot of data set curation as well. And I'll talk towards the end of the presentation about some examples of the types of projects that we've done. The type of approaches that we take are pretty much symbolized in the little example we've got on this slide. We curate data, build models, enable generation of predictions, and then perform in vitro experiments, and then ultimately in vivo, and feed the data back. And in the process of our work so far, we've been able to put together a number of collections of models, as well as develop a number of proprietary tools that allow us to generate new analogs of molecules, as well as perform retrosynthetic analysis. Some of the examples of software that we've been able to generate include our Assay Central software, which Tom will talk a little bit more about today. This is our core technology for data curation, as well as model building and validation. And in the process of using this technology, we've been able to come up with sort of spin-off products, including Megatox, which is a subset of toxicology models, Megatrans, which is a set of human drug transporter models, and finally Megapredict, which is our collection of thousands of models based on data sets that we've curated from Campbell. And this is also used for drug repurposing applications. Our technologies have allowed us to put together a pretty robust pipeline to date. And you'll see here that our Ebola and Marburg work is right at the top in terms of projects that we're working on actively. But we have quite a number of neglected and tropical diseases in our pipeline. And you can find more information about the type of work that we do on our website. So without further ado, I'd just like to give a brief introduction to Thomas Lane. Tom joined us in January 2018, but prior to that, he was a PhD student at UNC in North Carolina here, and he also did an internship with us in 2017 and did some work on our TB project. Currently at the company, he leads a number of our projects, including the Ebola project, our HIV project, and a DoD project on drug metabolism. And he's obviously going to talk today about the Ebola work. He's a pretty prolific author as well. So far, he's contributed to 17 papers at CPI. So without further ado, I'd like to pass you over to Tom. Thank you, Tom.
So thank you very much, Sean, for the introduction. So as Sean had mentioned, today I'm going to be focusing on the work that we've done over the last few years, which focused on finding therapeutics for Ebola. So just in general, I just wanted to briefly mention this presentation is going to be a synopsis of all the work that we've done over the last few years with regard to Ebola. For expansion, please refer to our publications or feel free to ask us directly. So I don't need to think I need to convince anyone that Ebola is still a real issue. In 2019, there was an additional outbreak which had over 2,200 deaths. So it's still something that's a real issue. And to date, there's only one approved therapeutic, which is a vaccine from Merck. So there are no approved small molecule therapies available for Ebola. So our involvement is trying to find small molecule treatments that actually began in 2015, as Sean mentioned, where we developed machine learning models to try to find potential therapeutics based on previously published in vitro data. These data included viral pseudotype entry and Ebola replication assays. So this was published in 2013. It was made up of 868 compounds. These were used to be able to create Bayesian models, which we then end up scoring, predicting activity from a microsource library, which consisted of over 2,500 compounds. So this is the general pipeline where basically we have a series of molecules. We assign molecular descriptors. We use structure-activity relationship to build models, and then we go to predict whatever library we wish, in this case, microsource, and then end up having some things to test. In this particular case, we ended up selecting 3 compounds, which was pyrineridine, quinacrine, and tillerone, and all of these ended up having nanomolar activity in vitro. Since these were compounds that were approved in at least 1 or 2 markets, this was conducive for drug repurposing. So as I just mentioned, we went from a high throughput screen, Bayesian models, virtual screens, and then in vitro activity. So the next step was going to be looking at in vivo activity. So we've looked at in vivo in mice for 3 of these compounds, all 3 of them, tillerone, quinacrine, and pyrineridine. Tillerone, we see 100% survival in mice, quinacrine 70%, and pyrineridine, we also see 100% survival. And this has led to 3 orphan drug designations for all 3 of these compounds. So as mentioned before, pyrineridine is going to be the focus of the rest of this talk. So in our pyrineridine studies in mice, we found that a single dose given IP of 75 mg/kg give 100% efficacy in mice infected with Ebola. We found that this also correlates with the blood plasma with reduction in viral load. This shows plaque assays and quantitative PCR. This was taken on study day 3, which was 3 days after the infection of Ebola. So we see a statistical drop in all the viral loads. We also did a short window study, which showed that we still get 90% survival after a 24-hour lag in treatment. This is not statistically different than the 100% because of the number of mice that we used. So it works with a little bit of a delay. We also have tested pyrineridine in a guinea pig model where we ended up looking to see with using an oral dose of pyrineridine, single 300 mg per kg oral dose ended up leading to a 40% survival in guinea pigs, which were infected with guinea pig adapted Ebola. So this was a small but significant increase in survival over vehicle. So this wasn't exactly what we expected. So we wanted to do a little bit more to be able to find out possibly why it wasn't as effective in guinea pigs. So our hypothesized reason, based on some of the in vitro data that we've gotten, is that the metabolic stability may be different in guinea pig as compared to mouse and also humans and non-human primates. So we actually do see a distinctive difference in metabolism, where we actually have a 2 to 3-fold lower half-life in guinea pig in microsomes, liver microsomes, that we have with non-human primate, human, and mouse. So it's metabolized much faster. We also see similar results in PK, where you end up having approximately 2-fold lower half-life in guinea pigs versus mouse in vivo. So we think that this may be one of the major issues with the effectiveness of this compound in guinea pigs. It's just because it just doesn't stick around quite as long. So it's less likely to be able to maintain therapeutic concentrations. So one thing that we've been pondering for a while is, how does pyrineridine actually work? So if we look at a very simplified pathway of Ebola, particularly the entry pathway, we end up having Ebola. It's particularly, I want to note that it does have a glycan cap, as this is something that's important for its ability to be able to enter the cell. We have various attachment factors, which then the cell will pick up the Ebola virus through macropinocytosis. And then we end up going through the endocytic pathway, which is common to many viral infections. So in the early to late endosome, which is about a pH of 6 to 5, we ended up having cleavage of the glycan cap, and this is known to be require cathepsin. And the next stage, which is late endosome, which is pH about 5, we end up having hemifusion of the Ebola virus, which now does not contain its glycan cap, and that ends up interacting with Niemann-Pick C1, which is a membrane protein found within the late endosome. So this essentially the next stage, which is thought to be likely to be a pH-dependent process, where you end up having complete fusion and then genome release into the cytoplasm. So since we know that cathepsin is responsible for priming Ebola, how do we end up using this to our advantage? So essentially, how do we end up increasing pH in the endosome and the lysosome? So one way to be able to do that is to look at lysometropic compounds. Now, this is in particular ones that are hydrophobic weak bases. So our example here is a lysometropic compound known as chloroquine. So this is in the cytosol in its base form, and it's also a protonated form. And it mostly exists in its base in pH 7.4. So this is because it's hydrophobic weak base, it ends up being able to diffuse into the lysosome. Once it's in the lysosome or late endosome, where there's a very low pH, you end up having a much higher concentration of the protonated base. And this actually makes it so it is unable to be able to get out of the lysosome. You have massive lysosomal accumulation, which theoretically can be thousands of fold higher. So this also leads to a pH increase, plus it's at a much higher level of the compound in the cytoplasm. So as I mentioned, Ebola entry does require cysteine proteases, cathepsins B and L, and these cathepsins are acid hydrolases, so their activity is pH dependent with an optimal activity of approximately pH of 4 to 5. So disrupting, by increasing the pH, we should be able to disrupt the function of this. So when going through to try to understand if this is something that we see in the literature, end up reading a lot of different papers and end up finding that there's a really strong relationship between lysosomotropic compounds and in vitro Ebola inhibition. So a paper that was published in 2011 in Toxicology in Vitro, they end up looking at different lysosomotropic compounds by looking at inhibition of lysotracker. And this was something that we know is a quantitative way to identify something that's lysosomotropic. Every compound that was less than 20 micromolar for IC50, we considered to be lysosomotropic. So we looked for an overlap for compounds that met this criteria that were also tested for Ebola inhibition. So 11 of these showed Ebola inhibition with an IC50 of less than 15 micromolar in vitro. And six of these compounds were actually tested against the Ebola pseudovirus and or VLP, and they also showed similar levels of inhibition, which definitely leads to understanding how Ebola is actually being inhibited, what stage, which is most likely entry. So this definitely strongly suggests that the lysosomotropic aspect of compounds is actually involved in the entry of Ebola. So this is available as a supplemental table in published work from this year. From us, it's a supplemental table if you want to go and look where some of these references and what came from. So from our perspective, we ask questions. So if we know lysosomotropic compounds are going to be something that is likely to inhibit Ebola entry, how do we end up finding these? So we use machine learning models to be able to do this. So we built a lysosomotropic model, which was built from model that was mined from the data literature. End up taking our CPI library and try to predict activity. And this is an example of things we looked at, particularly that were antimalarials. So chloroquine, artesunate, quinacrine, tilronine, and pyrineridine. We predicted all but artesunate to be active. This is the applicability score, which is basically how much overlap do we have with the model that exists. So we had looked at each one of these and looked at its ability to be able to inhibit a lysotracker, which we've found to be the quantitative way to be able to identify lysosomotropic so we see that chloroquine, artesunate follow the results that we expected. So artesunate was not lysosomotropic, while the other ones all were. So this model showed to be relatively, well, in this case, perfectly accurate. So as I mentioned, pyrineridine inhibits lysotracker, it's lysosomotropic. This is just an example of how we determine this. This was in a recent publication as well. So this was done in MCF7 cells, which is human breast cancer line. We saw that pyrineridine is not only lysosomotropic, but it's actually very potent, where it seemed to be the most potent of any of the published lysosomotropic compounds. Though I will mention that the lysosomal pH is actually cell dependent, and the more acidic it is, the will affect how much is inhibited. So this may be something that's cell dependent, but regardless, it is a very potent lysosomotropic compound. So we asked ourselves, is it just this lysometropic aspect of it, or is it something else that may end up driving its ability to even inhibit Ebola? So we performed MST experiments at pH 5.2 because we felt this was the most relevant, since this is essentially when you have cleavage. So the glycan cap was intact in this, so it would represent a slightly earlier stage before cathepsin is able to cleave the cap. So this was shown to be a stronger binder, pyrineridine, than all other published compounds, with the most potent ones before had a KD of approximately 25, which is trimethine. We have pyrineridine is about 7. So ask yourself as well, is there a relationship between how potent the Ebola inhibition is and both the lysometropic properties and the binding to the actual Ebola glycoprotein? So as I mentioned before, we know that trimethine was one of the most potent, one of the strongest binders, and it is actually presumed to be lysometropic, but the actual IC50 is not actually known, so we can't really judge whether this is going to be the case or not. But if we look at some of the other data that's out there for things that bind to Ebola glycoprotein, we see that you have several which are weak binders that are also lysometropic, and there is a correlation, which looks to be a correlation between both strong binding, lysometropic, in order to be able to be the best inhibitors. So we definitely think this is likely to be a relationship. So even though that these compounds have KDs that are may not seem relevant based on what we consider to be very potent, because these are lysometropic, we can have up to 1,000-fold increase in the lysosome over cytoplasm. So this is definitely something that would still make sense and be physiologically relevant. So the next, we looked at structural data to help elucidate the mechanism of pyrineridine. So we looked at crystal structures, which had inhibitors trimethine, ibuprofen, riperidil, paroxetine, and sertraline. And these all seem to be able to bind in the same region, which is an interface between GP1 and GP2. Interestingly, these compounds seem to destabilize the heterodimer by binding in that location. This is something that's previously published in a 500 micromolar. These shows a change of TM of -15, -6, and -3 degrees respectively. So it definitely appears to be destabilizing. We looked at nano-DSF to be able to confirm that these destabilizations also occur at the concentration we chose, which was 40 micromolar. And we also see that pyrineridine also seems to have a destabilization effect. So we believe that this is likely to suggest that similar mechanism. This is unpublished data. So now that we have basically think that there's similar mechanism, we asked ourselves, is it possible for this pyrineridine to share the same binding pocket? So we ended up doing docking studies, and pyrineridine definitely fits, and it picks up many interactions, which resulted in about -275 kilocalories per mole for binding. So it's definitely possible that it does fit in the same pockets. We are currently in the process of doing protein crystallization to confirm these docking studies with X-ray crystallography. So that is an ongoing process. So overall, going to give a little synopsis of the suggested mechanism of action. So we know that pyrineridine is lysometropic, so this would make it possible to be able to disrupt the proteolysis by cathepsin, which is pH dependent. We know that the pyrineridine binds directly to Ebola glycoprotein, which may affect the hemifusion of the glycoprotein with its receptor, which is Niemann-Pick. So this ultimately suggests that the pyrineridine is inhibiting the entry of Ebola And we have also shown this to be the case with the inhibition of a pseudovirus, which is VSV, which has been engineered to have the Ebola glycoprotein at the surface. So basically, we think that it is blocking entry, and this is due to its lysometropic character and also direct binding to the Ebola glycoprotein. So as really the real interest is to be able to bring it to the clinic to be able to help people suffering from this horrible disease. We wanted to be able to take our in vivo studies and translate that to clinical treatment of Ebola. So one question you want to ask yourself typically is that is the dosing that would be suggested something that's actually achievable? So the effective dose in mouse is 75 mg per kg, which end up giving 100% survival. This can be scaled to a human dose, which is approximately 6.1 mg per kg. So there are reported results for pharmacokinetics of pyrineridine in humans after an IV dose of 6 mg per kg, which was actually when it was used as an antimalarial. So therapeutic concentrations for Ebola treatment are achievable. So one of the caveats is that it failed to completely protect in the guinea pig model with 40% survival, statistically significant, but not as prolific as we could hope for. But there's data that suggests that this animal model may not be appropriate due to this drastic species-specific metabolism differences. So the next steps for this would be to test in non-human primates and hopefully bring it to the clinic with the animal rule. So as I mentioned, the whole idea of this is to be able to repurpose pyrineridine, and we've shown that it inhibits Ebola both in vitro and in vivo. We end up seeing the target is likely to be the glycoprotein for Ebola with a KD of approximately 7. We find that it's also lysometropic, which we suggest is involved with reducing the amount of cleavage and priming of the glycoprotein. So pyrineridine artesunate is something that's approved by the EMA. This is known as Pyramax. But there's a little bit more for pyrineridine that I hadn't mentioned yet, and then it actually is able to act as a monotherapy against parasites, viruses, and bacteria. So it's been shown before as an antimalarial that is potent against inhibiting malaria. It's also shown to have activity by Babesia, which is a equine disease. But we've also shown through multiple publications that it is active against not only Ebola, but Marburg virus, norovirus, this was found through an NIH screening, tuberculosis, and T. cruzi. And note that T. cruzi, we've also shown that it is effective in vivo as well. Something that I'm not able to disclaim today, but we also have found that it is active against another BSL-4 virus at the nanomolar level. So pyrineridine is a broad-spectrum antiviral, antiparasitic, and antibacterial. And we have orphan drug designations for pyrineridine for Chagas, 2 of them, Ebola, and for malaria as a monotherapy. So this work was not done by myself really at all. It was really just a huge team of people who were involved. Some of this work was done in the Davy lab, and we'd like to thank Rob and Menu for the work they did. This is in vitro. The Comer lab at UTMB, including Jason, Shane, and Alex. SRI, which was Southern Research Institution, Rutgers, and other CROs had done different things like chemistry and XPK pharmacokinetic consultants, and really Joel Freundlich and Peter Madrid. They were very instrumental for this whole project. We definitely like to also thank Julie Dial from the NIH, who ended up doing some work that was not directly in this presentation, which were some of the synergy studies and some other in vitro work. And I also want to have a special thanks to Minnie Davis from the NIH, who really helped this whole work to be pushed forward. And of course, want to be able to thank the funding sources, as without this, we wouldn't be able to have done any of this work. So without further ado, Sean.
Thank you, Tom. I'm just going to put the slides up, and thank you so much for the presentation. As you can see, Tom has brought all of the work that we've published together very nicely. I'm just going to take a couple of minutes to give you a little bit more information about the company. If you would like to take this time to write any questions you may have for Tom or myself, please again use the question chat box at the top or on the attendee chat within the software. We'd be happy to answer those questions in a few moments. As Tom said, we're very grateful for the support from the NIH, which funded not only the Ebola work itself, and that was funded by NCATS. And I'd like to thank Bobby Ann Mount for her support as program officer for that particular project. But also we have funding from NIGMS that supported the development of Assay Central, the software that was used for the machine learning, as Tom described. As a company, we, as I mentioned at the beginning, we do a lot of work around data set curation. And Tom mentioned that obviously for building the model for Ebola, we had to curate the data sets. Similarly, for the lysometropic activity, we had to curate and build those models. But we can certainly do this as a fee-for-service project if your company or your academic concern is interested in working on such projects. And really, the types of projects that we work on kind of range in size from relatively small data sets to massive data sets with hundreds of thousands of compounds in molecule libraries. We've got a lot of experience around prioritizing compounds for synthesis as well as testing. And we can certainly provide a ranked prediction for activity as well as ADME properties. We have considerable expertise in molecular property calculations as well. So that's something that we're happy to do, and we have different types of software to do that. And finally, as I mentioned at the outset, we've developed our own software for analog generation, assessing synthetic feasibility, as well as retrosynthetic analysis. And we're certainly happy to accept any new challenges that you may put in front of us. If you'd like to take a look at the Ebola computational model, we have it accessible at www.assaycentral.org. We have a number of our machine learning models there, as well as our COVID-19 models that we've been developing. In addition, we have several of our ADME models, P450 and HERG, as well as additional molecular properties. And we regularly update our COVID-19 viral models as well as our protease models. If you'd like to find more information about our software, we've published a number of papers initially describing the Bayesian approach as well as the ECFP6 fingerprints. And those papers are shown here. And obviously, most recently, we've published a number of applications of the technology, including the Ebola work. So as a company, why would other people like to work with us? Certainly because of the lack of angel funding and venture capital funding, we can pretty much choose the direction we want to work. And so we're happy to do fee-for-service work with companies or groups. And in these cases, the IP that we generate remains the IP of the company or the institute that's concerned. And we have no reach through. And so that's something I think that separates us out. We're happy to work on these types of projects. And we have a number of different client use cases. Very briefly, we've worked with consumer product companies, primarily working on endocrine disruption. We've worked with rare disease companies, as well as small animal CRO companies. And I think the thing that really ties all of these together is the close interaction we have with the organizations. And so there's a lot of back and forth in terms of sharing data and providing the models and the predictions that they need. So with that, I'm happy to answer any questions, and Tom is happy to answer any. So let's take a look to see if there are any that we can.
So I do see a question by Scott Miller, whether or not we've looked at amidiquine for the lyso tracker. So no, we did not. But based on the structure, I would feel pretty confident in saying that it is likely to be lysosomotropic. So maybe why that may have failed in non-human primate studies, and potentially why pyrineridine might. What we do know, pyrineridine also directly binds to the Ebola glycoprotein, and I'm not sure about whether imidaquine would or not. Plus, the lysosomotropic aspect is, just because something's lysosomotropic may not mean this accumulates quite as high in the lysosome, as that is something that is really very challenging to predict and really has to be looked at experimentally.
Thank you, Tom. We have a question from another of our attendees today. Do you see pyrineridine as a potential monotherapy for Ebola, or always paired with arsenate? That's a good question. I think that obviously it's already approved in Europe as a combination with arsenate as Pirimax. But I think from our side, it certainly has utility as a monotherapy. I think it could be potentially combined with another Ebola virus inhibitor. So, for example, remdesivir works by a totally different mechanism on the RNA polymerase. So I think maybe there might be benefits of using different molecules that have different targets. I don't necessarily, my personal opinion is I don't know that we necessarily have to go with Pirimax. I think that we've shown separately that obviously the arsenate has activity against Ebola virus as well. It's not as potent as pyrineridine, but I think based on the pharmacokinetics of pyrineridine, it's got a very long half-life. It's got low metabolism. It seems to be accumulated, so I think it's probably ideal.
Just to add, we did look to see if there was any synergy between arsenate and pyrineridine with an in vitro study that was recently published. While we didn't see official synergy, it did appear that it had reduced toxicity when given simultaneously. But we also did look at arsenate and find that it is not lysosomotropic. So if that is part of the mechanism, being lysosomotropic, they may be slightly different mechanisms, but as Sean said, there are a lot of advantages to pyrineridine as likely to be a monotherapy if it's effective, based on a lot of its pharmacokinetic properties.
One of the questions, actually it's a comment, is the dose for malaria in Pirimax is 2.1 grams pyrineridine split over 3 days. So that's a pretty short dosage even for malaria. But we would propose a single dose of pyrineridine itself as a treatment. And I think that's something that kind of makes it stand apart from other Ebola treatments that would probably have to be given daily and also IV. This would be obviously an oral treatment. And obviously a single treatment is much more convenient than having to take it every day for however long. So I think that's probably another differentiator. So happy to answer any other questions, but I would like to say if you have any other interest in learning more about what we do at the company, please visit our website. We've got a lot of information there about the types of projects and publications, as well as regular announcements on the types of webinars that we're putting out. You can also contact us directly. Our emails are shown on this slide. And I just have a little bit of a heads up that we're going to give our next webinar in about a month. We're going to focus on a presentation on our Alzheimer's disease projects. And that will be a webinar given by Patricia Vigneault, who's our new senior scientist. So that's coming up. And if there are no other questions, thank you all for joining us from all the different places in the world. We see we've got a number of attendees today from South Korea and other countries globally. So thank you all for taking the time out. And thank you for asking questions. And if you have any other questions, please drop us a line. Thank you. And thank you, Tom, for helping us today.
Thank you, Sean.